Welcome to This Week in Biochar, your regular update on the latest buzz from Biochar Today. We filter through the news and research so you don't have to.
That's right. We're here to give you the highlights, all AI generated, but with a human check to make sure it makes sense.
And this week we're diving into some pretty important topics. We'll look at best practices for lab work.
Making sure those water remediation studies are reliable.
Exactly. Then we'll shift to how biochar helps plants handle heavy metal stress.
A huge issue for agriculture and environmental health.
We've also got insights on biochar's effects in tricky soils, saline alkali ones.
And acidic soils, too, looking specifically at chili peppers in one study.
It really covers the spectrum from the lab bench to the field. Let's jump in.
Sounds good.
Okay, first up, that focus from Biochar Today readers on, well, quality control in the lab. There's a tutorial paper called Biochar-Based Contaminant Removal, a tutorial on analytical quality assurance.
Right. The goal here is pretty clear. Biochar research involves people from chemistry, engineering, soil science, all sorts.
So different backgrounds, maybe different standard methods.
Potentially, yeah. So this tutorial aims to improve the reliability, the credibility of studies looking at how well biochar sucks up contaminants, especially from water. It's about getting everyone on the same page.
Makes sense. What are the key things they emphasize for analytical quality?
Yeah.
Making sure the measurements are right.
Precisely. It starts with choosing the right tool. They mention things like atomic absorption spectrometry, AAS.
Good for looking at one metal at a time, usually.
Yeah, typically. Then there's optical emission spectrometry, OES, which is great because you can measure multiple elements simultaneously. Very useful if you have a mix of contaminants.
Okay.
And for molecular stuff like pesticides or dyes, UVV spectrometry is really common. If the molecule absorbs UV or visible light, you can often detect it that way. They also mention more advanced techniques like HPLC and LCMS.
So picking the right instrument is step one. What about using it correctly?
That's crucial. The tutorial stresses proper calibration curves. Think of it like ensuring your ruler is accurate before you measure anything.
Right.
They recommend using at least 5 standard concentrations, measured multiple times in triplicate and showing the error bars.
Yeah.
You need to know your margin of error.
And they mentioned the R-squared value, the statistical fit.
Yes, but they warn against relying only on R-squared. A high value doesn't automatically mean your calibration is perfect. Outliers, weird data points can mess things up. And also something called leverage points. If your standards aren't spread out well, a point far away can overly influence the line and make the fit look better than it really is.
So it's about careful design. What about sensitivity? How small a change can you detect?
That's basically the slope of your calibration curve. Sensitivity can be affected by things like the pH of the sample or the matrix, everything else in the water besides the contaminant you're measuring.
Matrix effects. That sounds tricky.
It can be. The matrix might enhance your signal or suppress it. So you could be under or overestimating the contaminant concentration if you don't account for it. They even provide an equation to quantify this matrix effect, M%.
Okay. And what if you have multiple contaminants that interfere with each other's signals?
That's selectivity. In UVVs, for instance, 2 different dyes might absorb light at similar wavelength.
Giving you a mixed signal.
Exactly. You might need to carefully select a specific wavelength where one absorbs much more strongly than the other. Sometimes chemical treatments, derivatization can help, but that adds complexity. Even in atomic methods like AAS or OES, you can get spectral overlap. Cadmium and arsenic sometimes interfere, for example.
Lots to keep track of just on the measurement side. Any practical tips for the actual analysis process?
Definitely. They suggest dual beam UVV systems are generally more stable than single beam. If you only have a single beam, you need to run your reference blank very frequently. Run your known standards regularly during a batch of samples to check for instrument drift. When you zero the instrument, use a solution that mimics your sample matrix, maybe even water that's been exposed to clean biochar.
To account for anything the biochar itself might release.
Exactly. They also recommend glass or quartz cuvettes over plastic ones, especially for organic compounds. Make sure sample and standard viscosities match for AAS, as that affects how they're drawn into the instrument.
And filtering.
Always filter samples before analysis to remove any tiny biochar particles. And choose your storage vials carefully, amber vials for light-sensitive compounds, specific plastics like polypropylene for things like PFAS.
That's a very thorough look at the analytical side. What about the actual sorption experiments mixing the biochar with the contaminated water?
Right, the batch sorption studies. It starts with storing the biochar correctly. Sealed containers, kept dry, away from air, ideally under an inert gas like nitrogen.
And maybe storing it in smaller amounts.
Good point, yeah. Store it in multiple small containers rather than one big one you keep opening. Particle size needs to be uniform, usually achieved by sieving. Smaller particles mean more surface area, but they can be harder to separate later.
Makes sense. And weighing it out accurately is obviously important.
Crucial. Using a calibrated analytical balance. You're often dealing with small amounts, maybe 10 to 100 milligrams.
And the contaminated water you create.
Use high-quality deionized or distilled water. Prepare a single stock solution of your contaminant if possible for consistency and verify its concentration. Consider the pH. Even CO2 from the air can dissolve and lower the pH slightly over time.
And the containers for the experiment itself.
Choose the right material: glass, polypropylene, Teflon, depending on what you're testing, to minimize the contaminant sticking to the container walls.
You need to check for that, right? Run tests without biochar.
Absolutely. You need blank controls with just the contaminant solution in the container to see if any is lost to the walls. Other controls might include the contaminant solution with the original biomass before it was pyrolyzed.
And keeping it all mixed during the experiment.
Continuous agitation is key. Shaking, stirring, vortexing. Different methods work. The important thing is consistency and finding an optimal speed. Too slow and mixing is poor, too fast, and you might break up the biochar particles.
And they mentioned temperature often isn't optimized.
Yeah, it's often just done at room temperature, but temperature can significantly affect sorption, so ideally it should be controlled and studied.
Okay, so after the mixing time is up, you need to separate the water from the biochar. How's that best done?
Filtration is common, but it has pitfalls. Some contaminants, especially dyes, can absorb onto the filter paper itself.
So you'd overestimate how much the biochar removed.
Exactly. Centrifugation is a good alternative. Spin the tubes so the biochar settles out, then carefully take the water off the top. Or you could pre-saturate the filter paper with the contaminant solution. Suction filtration or syringe filters can also be used, especially for fine particles.
Designing these experiments sounds systematic. How do they suggest approaching it?
They outline a structured process. Define your goal. Identify key factors: contact time, pH, initial contaminant concentration, biochar dose. Usually you vary one factor at a time, keeping others constant.
The OFAT method, one factor at a time.
Right. You measure the removal percentage, R%, and the amount absorbed per unit mass of biochar Q. They suggest aiming for R% between 20 and 80% for the most reliable modeling data. Less than 20%, and measurement errors become large, more than 80%, and it's hard to distinguish between different conditions.
So you'd test different amounts of biochar first.
Typically, yes. Vary the biochar mass while keeping the contaminant concentration and volume the same to find an optimal dose. Then, select an initial contaminant concentration, ideally one relevant to real-world pollution levels.
Then optimize pH.
Yep, test across a pH range, keeping concentration and dose constant. You have to be careful here, though changing pH can sometimes cause the contaminant to precipitate out of solution on its own or change its chemical form.
And finding the right contact time. How long to leave it mixing?
You set up multiple identical tubes and stop the experiment at different time points. You keep sampling until the concentration in the water stops changing. That's equilibrium.
And then you model this data?
Exactly. Kinetic models like pseudo-first order, PFO, and pseudo-second order, PSO, are commonly used. They can give hints about whether the absorption is more physical or chemical, though it's not definitive. There are other models, too, like intraparticle diffusion, to see if diffusion into the biochar pores is limiting the speed.
You need enough data points up to equilibrium, I guess.
Crucial. And the gold standard, though rarely done, is to actually analyze the biochar afterwards to directly measure how much contaminant it absorbed.
After kinetics, you look at isotherms, how much it holds at equilibrium.
Right. Here, you vary the initial contaminant concentration at the optimized biochar dose, pH, and contact time. You fit this data to isotherm models, like Langmuir or Freundlich. Langmuir assumes a uniform surface. Freundlich assumes a heterogeneous one. Biochar is usually heterogeneous, so more complex models are often needed.
Linear versus nonlinear fitting.
Nonlinear fitting is generally preferred nowadays, as linearizing can distort the error structure. You need to use appropriate goodness of fit statistics, like RMSE or at square, not just R squared, And critically, the parameters derived from the model should make physical sense. Don't just force fit the math.
They also mentioned thermodynamics, studying the effective temperature.
Yes, by running the isotherm experiments at different temperatures, you can calculate things like Gibbs free energy, enthalpy, and entropy using the Van Hoff equation. But they caution this needs care with biochar,
Why is that?
because changing the temperature might actually change the biochar itself, physically or chemically, and the assumptions behind the Van Hoff analysis might not perfectly hold. So it's useful, but should ideally be complemented by other techniques. They also stress a really important technical point about making the Langmuir constant dimensionless correctly before calculating Gibbs free energy, providing the right equations to avoid common errors.
Okay, and crucial for practical use. Can you reuse the biochar?
Hugely important for economics. The tutorial covers regeneration. Can you get the contaminant back off? Usually involves washing with a solvent or chemical reagent desorption. The best method depends on the biochar and the contaminant.
Stronger bonds are harder to break, I assume.
Generally, yes. Chemisorbed inorganic ions can be tougher to remove than loosely, physically adsorbed organic molecules. They give an example of challenges regenerating biochar loaded with phosphate. You need to measure the desorption efficiency and see how well the biochar works over multiple adsorption-desorption cycles.
And check if the biochar itself degrades.
Exactly. Monitor changes in surface area, pore structure, functional groups. You might also lose some biochar material during washing and filtering. There are other regeneration methods too, like using ultrasound or microbes, but chemical washing is common.
Finally, moving beyond lab water to real-world scenarios.
Yes, testing in real water matrices is essential. Consider the natural pH, temperature, presence of other ions, organic matter like humic acids. You can do this by adding competing substances to clean water, creating simulated natural water, or using actual water samples from rivers or wastewater.
Which brings its own challenges.
Right, filtration might be harder. Humic substances can interfere with UVV analysis. You might need chromatography or techniques like standard addition to overcome that. Standard addition is also useful in ASOSes with complex samples.
And looking at how different pollutants compete for spots on the biochar.
Also important, competitive sorption studies. You'd need methods like OES or chromatography that can measure multiple things at once. Usually best to understand how the biochar absorbs each pollutant individually first.
And the last point they mentioned is mass balance, trying to account for all the pollutant.
Yeah, it's ideal, but practically very difficult. Did it absorb to the biochar? Stick to the walls, degrade, volatilize? Doing a full mass balance under optimized conditions, though challenging, gives the most complete picture.
Wow, that's an incredibly detailed guide. Really underscores the need for rigor, as highlighted by the Biochar Today readers.
Absolutely. Getting these fundamentals right is key for building confidence in biochar as a remediation tool.
Okay, let's shift gears now to another area getting attention on Biochar Today. Using biochar directly in soil to help plants cope with heavy metal stress.
Right. This is based on a review paper titled Biochar Amendments in Soil, a Sustainable Approach for Mitigating Heavy Metal Stress in Plants. It tackles the big problem of soil contamination from things like industry and agriculture.
And these heavy metals hurt plants and can get into our food.
Exactly. They can stunt growth, reduce yields, and pose serious health risks if they accumulate in crops.
So how does biochar fit in here?
Biochar, made from pyrolysis heating biomass like wood or crop waste without much oxygen, typically 300–700 degrees C, creates this really porous, high surface area material.
We heard about that surface area in the water context.
Same principle applies in soil. That structure, along with chemical functional groups on its surface, allows biochar to absorb heavy metals. Mechanisms like hydrogen bonding, electrostatic forces, precipitation, they all play a role.
So it locks up the metals.
Largely, yes. It often has a high cation exchange capacity, CEC, meaning it can hold on to positively charged metal ions like lead, cadmium, copper, zinc, nickel, reducing their availability for plants to take up. This is called immobilization or stabilization.
The paper mentions specific metals.
Yes, it reviews studies for several key ones. For lead, biochar has been shown to reduce how much lead plants like ryegrass and maize take up and how much moves into the shoots of rice. It makes the lead less phytoavailable.
Can it work with other soil treatments?
Sometimes, yes. Combining biochar with chelating agents like EDTA might enhance phytoextraction using plants to remove the lead, but often the goal with biochar is long-term phytostabilization, just keeping the lead locked safely in the soil.
What about chromium? That one can be nasty.
It can, especially chromium-6, which is toxic and mobile. Biochar helps by binding it, sometimes reducing CR5 to the less toxic Cu and generally immobilizing it. Studies show less chromium in soil and plants like rice and tomatoes with biochar.
Combining it with other things helps here too.
One study mentioned combining biochar with elemental sulfur-enhanced maize health and chromium removal.
Okay, and cadmium, CD. That one moves easily into plants, right?
It does. It's quite mobile and readily taken up, which is a major food safety concern. Biochar helps by improving the soil's physical properties and, again, using its surface area and functional groups to sorb the cadmium, making it less available. Lower CD levels in roots and shoots of various plants have been reported.
Copper and zinc are micronutrients, but toxic in excess. How does biochar handle that balance?
It's a bit more complex. Biochar can reduce the bioavailability of copper and zinc, which is good for toxicity, but you wouldn't want to reduce it so much that the plant becomes deficient.
Right.
Studies have shown reduced copper uptake in sunflowers and lettuce, along with less stress. For zinc, biochar can lower its availability by increasing soil organic matter and changing pH. This has helped plants like rubber, maize, and tomato under zinc stress, improving growth and yield while reducing zinc toxicity symptoms.
Application method matters too.
Potentially. One study noted that integrating biochar into the soil versus just applying it on the surface affected copper uptake differently.
Arsenic is another really problematic one.
Definitely. Biochar application has been shown to improve growth and photosynthesis in rice while reducing arsenic accumulation in the grains. Similar benefits, reduced arsenic uptake, better growth, were seen in other plants like mountain brome and ryegrass.
Any particular biochar type better for arsenic?
Interestingly, one study highlighted that poultry manure biochar seemed particularly effective, not just reducing arsenic buildup, but also improving nutrient availability and plant antioxidant defenses compared to other biochars.
And nickel, also essential in tiny amounts, toxic in larger ones.
Correct. Nickel toxicity affects chlorophyll, growth, nutrient transport. Biochar's high CEC and surface area help immobilize nickel. Studies in wheat showed improved photosynthesis and reduced stress markers with biochar. Better growth and lower nickel levels were also seen in spinach and fenugreek.
So across the board, biochar seems to help plants deal with these metals. What are the common threads in how it works?
Several key mechanisms are highlighted. It often adjusts soil pH, usually increasing it, which can make many metals less soluble. It increases those negatively charged sites in the soil, CEC, that bind metal cations. Direct binding of metals onto the biochar surface is crucial.
Especially with some of the newer engineered biochars.
Exactly. Nano-engineered ones can be even more effective. Plus, biochar improves root development and water nutrient availability, making the plant generally healthier and more resilient.
And it helps the plant's own defenses.
Yes, it seems to strengthen the plant's internal defense systems against oxidative stress caused by the metals, boosting antioxidant enzyme activity and the production of compounds called phytokeratins that bind metals inside the plant cells.
It sounds like incredibly versatile. But the review also called for caution, right? Need for long-term studies.
Absolutely. While these short to medium-term results are promising, we need to understand the long-term persistence of these effects. How stable is the biochar? Does its effectiveness decrease over decades? Are there unforeseen impacts on soil microbial communities or nutrient cycles down the line?
Important questions for sustainable use.
Definitely. It's about maximizing the benefits while being aware of potential long-term consequences.
Okay, that provides a great overview of the heavy metal remediation potential. Now let's look at the last two pieces highlighted on Biochar Today, focusing on biochar's impact in specific challenging soil types: saline alkali and acidic soils.
Right, two papers addressing distinct soil problems. The first investigated how biochar amendment alters soil enzyme activities and microbial nutrient limitations in saline alkali soil.
These are soils with high salt and high pH, tough for crops.
Exactly. Salinization is a growing problem, reducing arable land worldwide. High salt causes osmotic stress, high pH reduces nutrient availability. This study used saline alkali soil from China and grew alfalfa, testing two different biochars, AL and AC, at different rates.
What were the main effects on the soil itself?
Good news mostly. Both types of biochar reduced the soil salinity, measured by electrical conductivity, and the pH. They also generally improved soil nutrient levels and boosted the alfalfa growth.
Some positive impacts on the basic chemistry and plant growth. What about the soil microbes and their enzymes? That sounds more complex.
It is. Enzymes are key to nutrient cycling. The researchers found quite different effects depending on the biochar type. AC biochar tended to suppress enzymes involved in acquiring carbon. Ale biochar, especially at higher rates, increased one carbon enzyme, glucosidase, but they both increased sucrase activity, another carbon enzyme.
And enzymes related to stress.
Both biochars reduced the activity of oxidative enzymes like peroxidase, suggesting less environmental stress in the soil.
They also looked at enzyme stoichiometry, the ratios between enzymes for different nutrients.
Yes, this gives clues about what nutrients might be limiting microbial activity. Ale biochar increased the CN enzyme ratio, suggesting microbes were investing more in getting nitrogen relative to carbon. AC biochar initially reduced it. Both biochars decreased the CP and NP ratios, suggesting phosphorus became relatively more limiting compared to carbon and nitrogen.
So the biochars shifted the nutrient landscape for the microbes.
That's the idea. Using something called vector analysis of the enzyme ratios, they found AL biochar clearly shifted the microbes from being primarily N-limited towards P-limitation. AC biochar showed a similar, though perhaps less pronounced, trend towards P-limitation.
Interesting. Did it affect how efficiently microbes used carbon?
Yes, both biochars increased estimated microbial carbon use efficiency, CUE. This suggests that a larger fraction of the carbon taken up by microbes was being incorporated into their biomass rather than being respired as CO2.
What drove these changes? Was it the biochar directly or changes in the soil?
Statistical analysis, redundancy analysis, pointed to different key factors for each biochar. For AL biochar, the addition rate, soil water content, pH, and ammonia nitrogen were important drivers. For AC biochar, available phosphorus and pH stood out. Interestingly, further analysis suggested that the biochar primarily influenced the enzymes indirectly through changes in the plant soil environment rather than direct effects.
And did the biochar change the types of microbes present?
Yes, and differently again. Ale biochar tended to increase bacterial diversity, while AC biochar decreased it. These shifts in the community composition likely contributed to the observed changes in enzyme activities and stoichiometry.
It really highlights how different biochars can act quite differently in the same soil.
Absolutely. The feedstock and production conditions really matter.
Okay, let's turn to the other study, corncob biochar in acidic soil used for growing chili peppers.
Right. This tackles the opposite problem: low pH soils. Acid sulfate soils, like those found in parts of Thailand where this study was done, can severely limit plant growth due to acidity and associated issues like aluminum toxicity.
And they use corncob biochar.
Yes, applied at a rate of 37.5 tons per hectare to reclaimed acid sulfate soil where chili peppers were grown.
That sounds like a lot of biochar. What did it do to the soil properties?
It made significant improvements. The pH increased, as expected. The cation exchange capacity went up, organic matter increased, levels of nitrogen, phosphorus, and sulfur rose, while potentially toxic soluble iron and aluminum levels decreased. Basically, it made the soil less acidic and more fertile.
And the impact on the chili peppers.
Very positive. The plants grown in the biochar amended soil had greener leaves, higher sp8 values, indicating more chlorophyll. Direct measurements confirmed higher levels of chlorophyll A, chlorophyll B, total chlorophyll, and also carotenoids.
Pigments involved in photosynthesis. Did that translate to better photosynthesis?
It seems so. They measured chlorophyll fluorescence parameters, which give insights into the efficiency of photosystem 2, PSII, a key part of the photosynthetic machinery. The results, like higher FEFM and eTORRC, lower DIRRC, indicated that PSII was working more efficiently, and the plants were experiencing less stress, dissipating less energy uselessly as heat.
Yeah, better growth and yield, too.
Definitely. Shoot length and overall plant dry weight were significantly greater in the biochar treatment compared to the control group without biochar.
And the important part, more chilies.
Yes, fruit yield, both fresh weight and dry weight, was considerably higher in the plants grown with corncob biochar.
So what's the connection? Better soil conditions leading to healthier plants.
Essentially, yes. By reducing the soil acidity and associated toxicity, like from aluminum, and likely improving nutrient availability, the biochar created a much better environment for the chili plants. The increased chlorophyll and carotenoids probably helped the plants photosynthesize better and also protect themselves from residual stress.
It counteracted the negative effects of the acidic soil.
Precisely. The study concluded that corncob biochar is a very effective strategy for improving chili pepper growth, photosynthetic performance, and yield in these challenging reclaimed acid sulfate soils.
These two studies together really paint a picture of biochar as a soil conditioner.
Yeah.
But its effects are clearly dependent on both the biochar type and the specific soil challenges you're trying to address.
Exactly. There's no single biochar solution. It's about matching the right biochar to the right problem.
So wrapping up our deep dive based on what's trending at Biochar today, we've covered the absolute necessity of careful standardized methods in lab research for water treatment.
Building that foundation of reliable data.
We've seen the broad potential for biochar to help plants withstand heavy metal contamination in soils.
A key role in remediation and safer agriculture.
And we've explored how different biochars interact uniquely with challenging soil types like saline, alkali, and acidic soils, affecting everything from soil chemistry to microbial life and ultimately plant productivity.
It really showcases the breadth and depth of current biochar research. Lots of aha moments connecting these different threads.
It definitely gives our listeners plenty to think about regarding sustainable agriculture and environmental protection, and maybe a final thought to leave them with.
How do we optimize biochar design and application to maximize the benefits for a given soil type or contamination issue while minimizing any potential negative side effects?
A complex challenge, but clearly where a lot of the research highlighted by Biochar today is heading. It requires understanding those fundamental properties and interactions we've discussed today.
Precisely. It's an ongoing journey of discovery and refinement.
Well, that's all we have time for on this deep dive from This Week in Biochar. Thanks for joining us.
Thanks for listening. We'll catch you next time.